That's a great question, and I think there are a number of advantages. So first, the personalized cancer vaccines that we've been seeing for the last decade, where people have had to have their tumor biopsies taken, they have to be sequenced, you have to identify all the potential neoantigens, then you have to make the vaccine. None of that is going to happen, I think, with the dark genome vaccines, because it appears that there's a limited number of these, and they appear to be shared. In a report that we had at SITC in November of 2025, we looked at 2 patients with head and neck cancer, and for the HLA-B44 haplotype, there were 70 dark antigens that were shared between these 2 different patients. And as we went to other HLAs, there were other numbers. They weren't quite as high as the HLA-B44, but it gives me great confidence that these dark antigens are going to be shared, because it appears that these dark antigens, in addition to being things that are not normally expressed in normal tissue, they're things that appear to have biological function that are driving the cancer malignant phenotype. And some really nice work from Prenzner's lab at University of Michigan, identifying in a recent paper 57 of these dark proteins, microproteins, that actually have tumor-promoting activity. So I think it's a really exciting time. The second part of your question is going to be, I think, translating this to the clinic, right? How are we going to do this? We've already been doing this with a biotech that we spun out, UBVac. They have a clinical trial with DPV-001, and working with Insight have, as I mentioned, I think a second ago, they've tripled response rates. So I think this is possible. I think it's possible to have things that are going to be off the shelf, and I think it's going to accelerate the clinical applicability and reduce costs, do a lot of other things that are going to be beneficial for the system.